I'd like to begin by thanking the organizers for inviting me to speak to you today at this Earl W. Davie Symposium about the research we've been doing in my lab on HDL signaling in cardiovascular disease. So I'll begin by acknowledging that the work that I'm going to tell you about was started by a number of former PhD students in the lab, Aisha Al-Jarala, Pei Yu, Leticia Gonzalez, and Christina Durham, and has also been carried on by a current PhD student, Alex Tian, and a current postdoc in the lab, George Klook. And we've also collaborated with a number of investigators. In particular, I'm going to point out Mansour Hussain and Abdul Momin at the UHN in Toronto. And my lab is funded by grants from the CIHR and the Heart and Stroke Foundation. So I'll begin with this diagram showing the main steps involved in the formation and metabolism of HDL. So HDL begins with the production of its major protein component, apolipoprotein A1, which is primarily synthesized in hepatocytes and secreted into circulation. And in circulation, ApoA1 can interact with cells and stimulate the efflux of phospholipids and unesterified cholesterol, which become associated with the ApoA1 molecules, forming discoidal nascent HDL particles. And these basically have the appearance of discs of phospholipid bilayers with cholesterol embedded, in which the apolipoprotein A1 molecules are wrapped around the hydrophobic fatty acyl tails of the phospholipids. So these can interact with cells, including macrophage foam cells in the artery wall, and stimulate the further efflux of unesterified cholesterol and phospholipid from these foam cells. And these become associated with the nascent HDL particles, and the unesterified cholesterol can be converted to cholesterol esters through the actions of an enzyme called LCAT, lecithin cholesterol acyltransferase. And the cholesterol ester, being much more hydrophobic, enters the core of the lipoprotein particle and forces the lipoprotein from a discoidal into a spherical shape as the HDL matures. Now, spherical HDL can continue to stimulate the efflux of unesterified cholesterol from cells, including macrophage foam cells, and this happens through the receptors ATP binding cassette transporter G1 and the scavenger receptor class B type 1. And mature spherical HDL can either exchange unesterified cholesterol and cholesterol esters with other lipoproteins through the actions of cholesterol ester transfer protein, or can carry its cholesterol cargo to cells where that cholesterol is selectively taken up via the scavenger receptor class B type 1. So in steroidogenic cells, such as adrenocortical cells, SRB1 mediates selective uptake of HDL cholesterol, and this cholesterol becomes esterified in the cell and is stored in cytoplasmic lipid droplets and is used as a reservoir of cholesterol for steroid hormone biosynthesis. In liver hepatocytes, the cholesterol is taken up by SRB1 into cells and can either be resecreted, repackaged into nascent lipoproteins and resecreted, or it can be secreted into the bile either as cholesterol or after conversion to bile acids and eliminated from the body. So this process, this net removal of cholesterol by HDL from macrophage foam cells and its delivery to the liver for secretion into bile is called reverse cholesterol transport, and is thought to be a major mechanism by which HDL protects against cardiovascular disease. And indeed, it's the most well-studied mechanism. Now, this is a diagram of SRB1, the receptor for HDL that mediates the selective uptake. And so selective uptake means that it's the lipid portion of the HDL particle that's taken up through SRB1 into the cell. And the lipoprotein particle itself is not internalized and degraded. And as you can see from the structure of SRB1, which was first described based on the solving of a structure of a related protein by Necrotic et al. And reported in Nature, this structure contains a channel which turns out to be a hydrophobic channel through which the lipids traverse from the bound HDL particle. And SRB1 can mediate the bidirectional flux of lipids from the bound HDL into cells or from the cell membrane out. Now, in addition to mediating lipid transport, SRB1 has been shown over the last decade to be able to mediate signaling or the activation of cell signaling pathways in response to HDL binding. Now, I'll point out this other protein that I'm showing on the diagram that's labeled as PDZK1. So this is an adapter protein that contains 4 protein-protein interaction domains called PDZ domains, the first of which binds to the carboxy terminal tail of SRB1. And in liver hepatocytes, PDZK1 is required to stabilize SRB1 against degradation. In other cell types, PDZK1 is expressed, but it doesn't stabilize SRB1 against degradation. Instead, it seems to be playing a role in this signaling pathway. And that's what I'm gonna talk to you about. Now, our interests in HDL signaling stem from our interest in the role of HDL in affecting macrophages responses during atherosclerosis development. So macrophages accumulate large amounts of cholesterol in the atherosclerotic plaque, and this cholesterol can become toxic as it accumulates in the macrophage and can induce cell death. And atherosclerotic plaques, in which there's abundant cell death of macrophages, develop regions that are called necrotic cores. So these are regions in the atherosclerotic plaque. They're characterized by evidence of abundant cell death. And atherosclerotic plaques with large necrotic cores and thin fibrous caps, such as the one shown on the top left, these atherosclerotic plaques are prone to rupture. And when a plaque ruptures, then the coagulation machinery is activated and a thrombus forms. And when this happens, this can fully occlude the lumen of the atherosclerotic plaque and result in a clinical event. So our interests are in what controls the cell death of macrophages and specifically how HDL might impact this process. So to begin to study this, we took macrophages from mice, put them into culture, and when we culture macrophages in lipoprotein-containing medium and expose them to a variety of stressors that trigger endoplasmic reticulum stress, such as tunicamycin, we saw, as others had reported, that macrophages are very resistant to cell death induced by endoplasmic reticulum stress. However, when Pei Yu in the lab depleted lipoproteins from the growth medium, she found that tunicamycin or the macrophages became much more sensitive to tunicamycin-induced cell death. In this case, she measured apoptosis using tunnel staining for DNA fragmentation. And as you can see, there's a large abundance of green tunnel-stained macrophage nuclei. So quantifying this, again, she saw tunicamycin-induced cell death in macrophages in the absence of lipoproteins. And when she added back different lipoproteins, she found that HDL could attenuate this tunicomycin-induced cell death in a dose-dependent manner, whereas other lipoproteins such as LDL could not. When she looked at macrophages that lacked PDZK1, the adapter protein that binds to SRB1, she found that HDL was unable to suppress tunicomycin-induced cell death. And similarly, Aisha El-Jarala, another former PhD student in the lab, found that when SRB1 was knocked out in macrophages, HDL was unable to protect them against cell death. So Pei Yu went on to look at the level of AKT phosphorylation in wild-type macrophages treated with HDL, and saw that, as others had reported in endothelial cells, incubation with HDL increased phosphorylation of AKT. And this increase in phosphorylation was lost when either PDZK1 was knocked out, or when AKT1, one of the three isoforms of AKT that's expressed in macrophages, was knocked out. So this suggested that this increased AKT phosphorylation represented AKT1. And when she looked at AKT1 knockout macrophages, she found that HDL was unable to protect them against tunicomycin-induced cell death, suggesting that this process involved signaling via AKT1. So she was interested in seeing whether this played a role in vivo in atherosclerotic plaque formation. And to do this, she took LDL receptor knockout mice and transplanted them with bone marrow, either from wild type or from PDZK1 knockout mice. When she fed the mice high-fat, high-cholesterol diets to induce atherosclerosis, she saw an increase in atherosclerotic plaque size in the mice that lacked PDZK1 in their bone marrow, and a much more pronounced increase in the size of necrotic cores in the atherosclerotic plaques of the mice that lacked PDZK1. When she looked for evidence of apoptosis within the plaques, either by tunnel staining or looking at activated caspase-3, a critical caspase in apoptosis, she saw increased evidence of apoptosis both by tunnel staining and caspase-3 labeling in the plaques in mice that lack PDZK1 in their macrophages. So this suggested that SRB1 and PDZK1 played an important role in mediating HDL signaling, leading to protection of macrophages against atherosclerosis. Now, in other research that I'm not going to show you the data of, but I'll just summarize, we've also been interested in this G protein coupled receptor called the sphingosine-1 phosphate receptor 1. And that's for a number of reasons. One is that HDL is known to be a carrier of the ligand for the S1P receptor 1, and that ligand is sphingosine-1 phosphate. And others had shown that SRB1 and the sphingosine-1 phosphate receptor 1, at least when overexpressed in cells, could associate physically. So we were wondering whether S1P receptor 1 might be involved in this HDL-dependent signaling pathway. And one of the main reasons is that S1P1 is known to induce a variety of signaling pathways, including PI3 kinase and AKT phosphorylation. So Leticia Gonzalez generated S1P receptor 1 deficient mice and did the same types of experiments that Pei did, transplanting bone marrow from S1P1 knockouts into LDL receptors, and saw, like Pei showed with PDZK1 knockouts, that those mice developed increased atherosclerotic plaques with larger necrotic cores and evidence of more apoptosis within plaques. So we think that this SRB1 and PDZK1 are working together to mediate HDL signaling. Now, as to what the role of PDZK1 is in this, we're not sure. One of the things that we hypothesize is that PDZK1, by virtue of its multiple protein-protein interaction domains, may actually be involved in the association of these two receptors with each other. And that's something that we're pursuing. Now, we're also interested in this signaling pathway in other cell types, and in particular in cardiomyocytes. And that's because evidence over the past decade or decade and a half have shown that HDL can have direct effects on the heart itself. So Christina Durham in the lab looked at cardiomyocytes from either wild type or SRB1 deficient neonatal mice, or immortalized human ventricular cardiomyocytes, and demonstrated that the wild type cardiomyocytes and the human cardiomyocytes express SRB1. And you can see in the SRB1 deficient cardiomyocytes, there's no SRB1 signal. So she wanted to look at whether HDL could protect cardiomyocytes against stress-induced cell death. And she chose to look at doxorubicin, which is a chemotherapeutic agent used to treat a variety of cancers, but which has a major side effect of cardiotoxicity. So in patients treated with doxorubicin, many years after doxorubicin use, they tend to develop cardiac damage leading to heart failure. So this involves a weakened left ventricle, which means the left ventricle cannot pump properly. And this is shown, or this can be measured by measuring the rate of change in pressure as the left ventricle contracts. In a normal heart, this rate of change in pressure is high. When the heart is damaged because of cardiomyocyte cell death, the left ventricle cannot achieve such high pressures during contraction. So she first put cardiomyocytes in culture, treated them either with HDL or without HDL, and exposed them to doxorubicin and measured apoptosis by tunnel staining. And as you can see, doxorubicin increased cardiomyocyte apoptosis. HDL pre-treatment of the cardiomyocytes attenuated this when the cardiomyocytes were from wild-type mouse hearts. But when the cardiomyocytes were deficient in SRB1, HDL was unable to protect them. Looking in vivo, she wanted to use a system where she could manipulate HDL levels. And as I mentioned to you before, HDL production is driven by the production of ApoA1. So she used ApoA1 transgenic mice that overexpress human ApoA1, leading to an increase in the amount of HDL in circulation. And so she treated these mice with doxorubicin, 5 milligrams per kilogram per week for 5 weeks, and then analyzed them a week later. When she looked at the apoptosis in cardiomyocytes in sections of the heart, she saw that in wild-type mice with normal levels of ApoA1, doxorubicin increased cardiomyocyte apoptosis, quantified here. But when ApoA1 was overexpressed, doxorubicin induction of cardiomyocyte apoptosis was attenuated. When she did the same experiment in ApoA1 transgenic mice or mice with normal levels of ApoA1, but which lacked SRB1, she saw that although doxorubicin increased apoptosis in these mice, ApoA1 overexpression was unable to protect them against this increase. And when she measured left ventricular function, she saw that in the wild-type mice, doxorubicin reduced left ventricular function. This reduction was attenuated when mice overexpressed ApoA1 and expressed the HDL receptor, SRB1. But when HDL receptor was knocked out in the mice, ApoA1 was no longer able to attenuate the doxorubicin-induced decline in left ventricular function. And then she and postdoc George Kluck went on to show that they got the same results when they injected mice with purified ApoA1 rather than using an overexpression model. So in conclusion, in addition to protecting against atherosclerosis-induced cardiovascular disease, HDL signaling appears to play an important role in protecting cardiomyocytes against cell death. and suggesting that HDL can protect either directly or indirectly against cardiovascular disease. So HDL does more than just transfer cholesterol out of the heart or out of vessels, but has direct effects both on the cells within the atherosclerotic plaque, as well as cells in the heart itself. So with that, I'll end my presentation, and I'd be happy to take questions.
Thank you very much. Serious feedback. Thank you very much, Dr. Trigatti. That was very interesting. I have a question for you. So I'm just curious about whether you think your next step is to go into human trials with the doxorubicin ApoA1 story.
That would be very interesting. I think that before we do that, we'll have to do a fairly careful look at other tissues, in particular in the liver and in kidneys, as well as move this into a tumor model to show that HDL is not, as it's protecting against doxorubicin-induced cardiotoxicity, that it's not also diminishing doxorubicin's ability to kill tumor cells. So I think we have to do those first before we can move into human trials or towards those types of studies.
Yeah, thank you. That makes lots of sense. Other questions in the audience? Dr. Pricedale.
So I don't know if a question was asked. I couldn't hear anything.
This is yes. Can you hear? Can you hear this now?
I can now. Thank you.
Probably a huge echo. It's Pricedale here, Dino. Nice to see you. Nice to see you, I guess. Yeah, I'm sorry you can't see me. I thought that maybe we would be projected in your Zoom, but in the audience here. But I guess you can't see us. You can't see this motley crew. Thank you very much. Everybody knows about foam cells. So how is this signaling pathway contributing to foam cell development? And maybe as really one of the first steps, can we start turning off that first?
So as I understand it, the question is, how is the signaling pathway, or is this signaling pathway contributing to foam cell development? And we had done some early studies looking at SRB1 knockout macrophages and PDZK1 knockout macrophages, and found that when we treated those macrophages with oxidized or modified lipoproteins, such as acetylated LDL, we really saw no difference in foam cell formation. However, when we treated those macrophages simultaneously with HDL, we could see that caused a reduction in foam cell formation. But that we're not sure if that's because of HDL effects activating signaling pathways, or whether that reflects HDL's ability to remove cholesterol from cells that the oxidized or acetylated LDL is delivering to cells. So it's unclear whether the signaling pathway may pardon me for that, may impact the foam cell formation, or whether that may be more of a reflection of HDL's cholesterol efflux capabilities. But those are things that we have to return to, I think, and look in more detail at.
Okay, the signaling pathway is fascinating, completely new to me. Have people tried to sort of extend this in a sort of more clinical sense, in that people with high level of protective HDL but advanced atherosclerosis, do you anticipate finding genetic abnormalities in your signaling pathway?
Look for that. One thing that I'll mention is that the extent of elevation of HDL that we achieve in the mouse studies with the ApoA1 transgenics, I think far exceeds the level of increase in HDL that you'll see or the range of variations in HDL that you see in human populations and individuals. So it is a bit of an artificial system. Having said that, though, there have been reports of individuals with very rare inactivating mutations in SRB1 in the receptor. None to my knowledge about individuals with mutations in PDZK1. But in those individuals with rare mutations that inactivate SRB1, there have been reports of a substantial increase in susceptibility to coronary artery disease, suggesting that, whether it's, again, it's hard to tease out the role of SRB1 in the signaling versus the reverse cholesterol transport pathway. But certainly it speaks to an importance in that receptor for protection against coronary artery disease.
Oh, it's Ed Conway. Thank you very much. That's a great talk. I had a question about the role of HDL and its effects in terms of signaling via adipocytes. Is it the same pathways involved? I understand it has some anti-inflammatory properties, but what other pathways are involved?
Yeah, so we haven't studied adipocytes ourselves, but this PI3 kinase AKT signaling pathway has been reported in a variety of different cell types, and including adipocytes and endothelial cells. Whether the sphingosine 1 phosphate and the sphingosine 1 phosphate receptor 1 play a role in that, I'm not sure.
Just a reminder to everybody attending that they can send in their email in your questions during the symposium. Dana, I'm going to hand it back to you.
Great. Thanks very much, Dr. Trigatti, and thank you.